Haruko Obokata was a stem cell researcher at the Reagan Center in Japan. In 2014, she published 2 journal articles in the Nature Journal, which were titled Stimulus-Triggered Fate Conversion of Somatic Cells into Pluripotency and Bidirectional Development Potential in Reprogrammed Cells with Acquired Pluripotency. In these articles, she stated that she was able to acquire pluripotent stem cells by simply submitting somatic cells in a citric acid bath. These were named Stimulus-Triggered Acquisition of Pluripotency Stem Cells. This was a huge discovery in the stem cell field because these pluripotent stem cells could then be differentiated into any cell in the body. This would allow us to generate tissue or organs which will have 0 chance of getting rejected by the body because they're made with your own cells. After the articles were published, many questions started to arise. The images seemed modified. The experiment was not able to be replicated. And there were parts of the article that seemed to be directly taken from other articles without being cited. Nature decided to retract the article until the experiment could be replicated, and Reagan Center started an investigation on Obokata. The investigation concluded that, in fact, the images were modified. For example, here's an image from the journal article titled Stimulus-Triggered Fate Conversion of Somatic Cells into Pluripotency. In figure 1F, the D2 and D3 was proved to have a artificial coloring of the cells. In the section I of the figure, lane 3 of this gel was found to be added later on, as for this lane belongs to a different gel. And for the second figure, this is figure 2 of the same study, this was found to be the images she used in her doctoral thesis, in which she submitted bone marrow cells into a thin tube and to create spherical cells through mechanical stress. Now, as you can see, this has nothing to do with the stem cells that she was trying to create in this other experiment. As for the replication of the experiments, they found that Obokata only had 2 journals of the 3 years of research that she had done. So the replication of the experiment, it was impossible because the notes were not detailed enough to replicate the experiment. And the investigation concluded that Obokata was guilty of research misconduct on various accounts through image manipulation and using images from other experiments without citing them. On her methods section, where she explains karyotyping, she directly copied 17 lines from a different journal article, and she did not cite this. When Obokata was questioned about this, she explained that she simply forgot to cite it and that she didn't have the source from which she copied it. And for the image manipulations or the images taken from her doctoral thesis, she stated that they had a similar name on her computer and that was just a mistake. However, this was not enough to justify the research misconduct, and she was still found guilty for image manipulation of the gels and for plagiarism. Now, Obokata could have avoided this research misconduct simply by citing her images that she used from another experiment, as this is a form of self-plagiarism. She should have cited other authors' work. And as for the image manipulation, it just simply should not have been done. Research misconduct directly obstructs the advancement of knowledge, which contradicts the goal of any scientist. Research misconduct is unethical because it betrays the trust of the scientific community and of the public.